course, have such a small wavelength that they have high resolution atomic scale. Then when they strike the crystal, they diffract around it, and they strike film, and then you get an image on a film after you develop the film, and you can measure distances between these spots. Okay, and this is one of the clearest photos she took. Now, this is the photo that Watson and Crick used to deduce the correct structure, but they were given the photograph without her permission. So that essentially it was stolen data. So very unethical. Because, as I said, Wilkins and Franklin didn't get along. Wilkins took the photograph up to Cambridge and gave it to him without permission. So she really got the shaft on that. But what they could tell from the photograph, the width of DNA, okay, and these spacings here suggested something like a spiral staircase. Now, just a few years prior, I talked about their main competitor was the guy at Caltech, Dr. Linus Pauling, who was also one of the, he had a Nobel Prize already on understanding atomic structure, and he was one of the first to use this technology to deduce the protein structure. He was able to identify the structure of collagen, which has a helical shape. So it was something in their minds. Okay? And Pauling, as I said, was working on the project, and his son was at Cambridge writing his dad letters about what these guys were doing. So he's like a little spy there. Okay? That deep state at Cambridge. And but Pauling didn't know this photograph. He had the wrong width. Okay, he thought that it turned out he thought it was 3 strands wide, but Watson and Crick were able to deduce it was 2 strands wide. And the spacing between these spots here, they were to deduce the distance between a nucleotide and able to tell how many the helical shape might rotate in one complete turn, and how many nucleotides you get back in one turn. So they got a lot of details about the dimensions of the molecule in this photograph that they obtained in an unethical manner. Well, they started to build these structures, and the first one of the early ones they built was totally wrong. They organized it where they put the sugar phosphate backbones. They knew it was 2 strands wide from the dimension of the photograph. They said, okay, we got this double-stranded molecule. It looks like it's a helical shape, but it's something we're all kind of thinking about now. So let's put but they weren't sure sort of the organization of the molecules. They put of the 2 strands, of course, base here represents the A, T, G, or C. They put the sugar phosphate backbones, the 2 strands, next to each other. They built a molecule. They built a model, and they invited Dr. Franklin up to see it. And she thought, you guys are like idiots. Again, you've got to understand where she comes from. She's been abused continually in her career, especially by Wilkins. And turns out Watson was kind of a sexist and stuff. He actually lost his job later in his career because of sexist comments he made. And they finally said, get that hell out. We don't care if you got a Nobel Prize. So when she saw it, she goes, you guys are like cowboys. I'm not sure. I think she used some more descriptive terms. I'm not sure. I'm not sure what an English woman would say to a male. She was she reminds me of, if you remember the show Seinfeld, anybody ever see that? There was the character Elaine, if she got pissed, she'd beat the hell out of everybody in the set. So she was like the Elaine of biochemistry, slam these guys against the wall. But obviously this can't work because phosphates have negative charges, and how are these negatively charged things going to hold each other together without propelling apart? So she looked at it and walked out, and that's when the director lab said, you're off the project, you've embarrassed us. So again, a lot of the work was done at the pub there on cocktail napkins. So they rearranged it, and they put the sugar phosphate backbones on the outside with the bases facing inward like rungs of a ladder. That part they got right. So they rearranged the molecule. Let me have a sketch of that, a slide of that, so let me just so you understand what I mean. So her critique, it's good to listen to criticism, even if it's acidic, because you might learn something. So they realized, okay, the sugar's phosphate backbone has to be on the outside so that the negatively charged phosphates don't repel each other. Okay? But if you got these negative charges want to repel each other, you got to have something to hold the 2 strands together. They figured, okay, the bases attached to the sugars are holding it together with some force that's counteracting this negative charge repulsion. And that DNA is like that. You got the sugar phosphate backbone have repulsive forces wanting to pull the double helix apart, and the bases are holding it together. Okay? And they're connected to the sugars. They figured, okay, so the bases are, I'm trying to line this up as best I can. So something between the bases, some force between the bases, we know now it's hydrogen bonds, is holding the 2 strands together. They're on the right track, okay? But they weren't sure how do the bases pair up. They weren't sure about that, okay? So at first they thought, well, maybe it's, remember there's 4 bases. There's A, there's T, there's C, and G. Okay? And we'll look at the next slide, but they come in 2 classes of chemicals. The 2 of them are called purines, that's the A and G. And purines have a double ring. Okay? And then the C and the T are called pyrimidines. There's different types of ring structures with nitrogen in them. And that's the C and the T. And that's a single ring base. Okay? So their initial attempt was, let's pair them up like with like. So I'll go ahead and write that in here. That means they thought, well, if you've got an A here, you got an A there. You got a T here, you got a T there. You got a C here, a C there, and a G here, and a G there. I think I got them all 4 there, right? Okay? So that was their first attempt. As they looked at the model, so here's the idea. You pair like with like. 2 double ring purines together, single ring pyrimidines together. And they realized, wait a minute, this isn't going to work. Because we go back to the photograph from Dr. Franklin. I like to look at her beautiful face. The molecule had a constant width all along the double helix. And if you pair like with like, the molecule is going to get fat and skinny and fat and skinny. Does that make sense? So it ain't going to work. So pairing like with like is no good. Okay? All right? Well, part of the problem was that Watson had the chemistry of the bases slightly wrong. So a colleague, another grad student there, said, hey, chump, this is the right structure. So they changed the structure of the base, okay? And they begin to fit together. That if you have a double ring A here, it'll pair with a single ring T. If you've got a double ring G here, it'll pair with a single ring C. Okay? And that'll keep the molecule at a uniform diameter, single ring to double ring. And the way they were able to figure out that A goes with T and not with G, because these are the double rings, was the data from Chargaff's paper published in 1950, where he was comparing DNA from different organisms. Here's the table from here's Erwin Chargaff, a dashing young Austrian. He had extracted DNA from these 6 species. 2 bacteria, E. coli. This is a strain that's fairly non-pathogenic, that's widely used in cloning today. This is a bacteria that causes tuberculosis. There's a single-celled eukaryote, a fish, a rat, and a rat. I'm just kidding. What he'd done is he'd taken DNA from these different organisms and boiled the hell out of it to break the polynucleotide chain into individual monomers, individual nucleotides. Then he developed a paper chromatography method where you then put the sample at the bottom, let it climb up the paper, and it separates into the 4 different nucleotides, because they have different polarities. And then you could quantify the presence of each one, do some sort of wet test. So you could quantify the percent composition of the 4 bases from each of these species. And he found 2 results. One, he kind of expected, the other one was bizarre, they couldn't explain. The first one was that since Avery's paper 44 had said DNA is a genetic molecule, and he was looking at his DNA from different species different, you would expect that this is a genetic code that would be different between different species, right? You'd expect our genetic code to be different from a bacteria. You would expect it, right? And that's what he saw. He saw that the percent of these bases weren't the same amongst different species. I mean, look at the percent A just of that between these 2 bacteria. It varies all over the map. So different species had different DNA composition. So somewhere the code is embedded there. The molecular code of inheritance is embedded somewhere. We can see it. We can't see the code here, but we know that the composition of DNA between different species is different. Okay, that made sense to Chargaff. What didn't make sense was that he noticed that in most cases, the percent A always roughly equal to percent T, and the G equal to C. Now it's a little off here. His methods were crude, but roughly, if you look here in an organism, and it should sum to about 100%, but if you have roughly 25, 26% T, 25 same, those 2 are the same, these are about the same, these 2 are the same. So the AT percent A equal to percent T, and the percent G equal to percent C. And that became known as Chargaff's rules, but he didn't know what it meant. But Watson and Crick said, ooh, that's the clue. Since we're trying to pair a double ring purine with a single ring pyrimidine, if A always equals T, that's 2 pairs together. Because as long as you got an A, you got to have a T. They're always going to be about the same. So that was the clue that allowed them to identify what's called the complementary base pairs. Does that make sense? Based on Chargaff's data. Okay? Complementary base pairs. And once Watson had corrected the chemistry of the nucleotides, he saw that if you paired an A with T and G with C, that the geometry would line up such that they could perfectly hydrogen bond to each other and not to the wrong base. And this would be the force that would hold, the attractive force that would hold these two strands together that want to repel each other because of the negative phosphates in the backbone. A forms 2 hydrogen bonds with T. G forms a stronger connection, 3 hydrogen bonds between G and C, and this match just won't hydrogen bond. They don't have the proper geometry. Okay? So that was the final piece of the puzzle for Watson and Crick. So they wrote up the results in a 2-page paper to the leading scientific journal in the world, Nature. This is the second page is just references, so I didn't include that. This is their 1-page paper that they got them a Nobel Prize. Now, who got the Nobel Prize? Watson, Crick, Wilkins. That's it. What about Dr. Franklin? It was her data that really turned the tide. Because she was a woman? I don't know. What the Nobel committee has always said is that you have to be alive to get the Nobel Prize. She died of ovarian cancer before the prize. Because we worked with x-rays our whole life. And people didn't understand the danger of x-rays. She actually was hiking in Mount Whitney.